Welcome to this walkthrough video on how to access recognized conformational states from the PDB in Europe knowledge base. Because proteins are dynamic macromolecules that typically adopt different shapes, otherwise known as conformational states, they control many cellular processes and often drive disease progression. Therefore, we have developed a software tool to cluster protein chains in the PDB archive based on their atom coordinates. To access this information, first head over to the PDBe-KB's website, linked in the description, and search for a protein chain by uniprot accession or PDB ID. I'll be using the uniprot accession for adenylate kinase in this example. You can visualize the clustered chains by clicking on 3D view of superposed structures at the lower left of the page. This will open a Molstar window, allowing you to rotate, translate, zoom, and clip the superposed protein chains. You may also adjust how the protein chains are rendered under the Component tab on the right-hand toolbar. In the center view, single protein chains with complete sequence identity are structurally aligned or superposed in order to visualize the differences and similarities between structures of the same protein in the PDB. The left-hand toolbar displays structurally similar groups of protein chains labeled here as clusters. Each cluster represents a set of protein models from the PDB archive that have model coordinates more similar to each other than those in the other cluster. Some proteins are grouped into several clusters if lots of conformational heterogeneity is captured by the entries in the archive. Therefore, these clusters represent possible conformational states. Only one representative chain is displayed per cluster by default. To reveal more chains, scroll or search for your PDB entry and click the eye icon next to the PDB label. All chains can be revealed and hidden along the top of the cluster label. To account for possible discontiguity, or in other words, protein chains in the PDB that match different regions of your queried uniprot sequence, you can toggle between these so-called segments at the top of the left-hand toolbar. If you would like to access the rotation translation matrices used to align the chains in 3D space, or the dendrograms demonstrating how clusters were determined, click on the Superposition Data button at the top right of the page. Cluster annotations can also be accessed by querying the PDBe's Graph API. For more information on how the clustering pipeline works, click on the documentation button. Predicted protein structures from the AlphaFold database can now be loaded into the Molstar viewer as well, allowing you to visualize the experimentally determined and predicted protein models in one place. Simply click the Load AlphaFold Structure tab, wait for the model to render, and now you're set to begin assessing how the structures compare. The right-hand toolbar will fill up with a new slider to toggle PL DDT cutoff and opacity, as well as a heat map of predicted aligned error, or PAE, extracted from the AlphaFold database. Noteworthy is the reported RMSD values between the AlphaFold structure and the representative models for each recognized cluster, clearly highlighting which potential conformation the predicted model adopts. Finally, you can also view our recognized protein conformational states along with their bound ligands. Return to the summary page and click the teal button titled 3D View of Superposed Ligands. An almost identical Molstar window will open, with many of the same features as before. However, hiding and revealing PDB entries on the left-hand toolbar will now toggle the visibility of the protein's bound ligands. This can also be achieved, but for all copies of a given ligand, by toggling the visibility on the right-hand toolbar. Thank you for watching this video on how to view and access the PDBe-KB's recognized conformational states. We hope these tools aid in your research and always value hearing your feedback.